So I'm Richard Tothill. I'm the head of the Rare Disease Oncogenomics Group at the University of Melbourne Centre for Cancer Research. It's my great pleasure to introduce Andrew Pattison from my group. Andrew is a very important member of my team who has been working in my lab for more than 2 years. Andrew is a postdoctoral scientist. He's also a bioinformatician. He has a few great qualities. For one, he's a very nice guy, if you know him. You would probably agree with me. He's also shown a great proficiency in bioinformatics and has shown great adaptability also to analysing many different types of data. He's also a very good teacher, and he's been really important for training young people in my lab on the use of bioinformatics, using command line bioinformatics and analysing different types of data. So today he's sharing with you some details about his experience in using single cell or applying single cell RNA sequencing, and that's to a rare type of a rare neuroendocrine tumour called a Phaeochromocytoma. This is a project that's been running for quite some time with significant input from others, but Andrew's made a massive contribution in the last 6 months to this work. So it's now coming to its natural conclusion, and hopefully we should have a manuscript prepared pretty soon. So without further ado, I'll hand over to Andrew.
Thanks very much for that very kind introduction, Richard. Thanks to Zoe for giving me, on the UMCCR, for giving me the chance to speak. So today I'll be talking about single cell and bulk gene expression analysis of rare neuroendocrine tumours. As you can see by my background, I'm going to do this with some help from the movie The Matrix. There's lots of parallels that can be drawn in this sort of analysis. Okay. So yeah, the main things we're going to be talking about today, I will introduce my talk. I will talk about single-nuclei RNA-seq, how you can analyse it, trying to determine cell type specific subpopulations from the single-nuclei RNA-seq data. Then I'm going to introduce a concept called pseudobulk of single-nuclei RNA-seq. And lastly, I'm going to talk about cell-to-cell signalling and theranostic targets from this data. So let's talk about that when I get up to it. Okay, so the disease, Phaeochromocytoma and paraganglioma. They're rare neuroendocrine tumours, and they arise from cells called chromatin cells that are associated with the sympathetic nervous system. This is the part of the nervous system that's responsible for the fight or flight response. These tumours are very rare, and about 10 to 15% of them go on to become metastatic. So this chromatin cell type secretes catecholamines, such as like adrenaline or adrenaline dopamine, again, responsible for that fight or flight response. The tumour is kind of grouped into a few different locations. So most PPGLs are phaeochromocytomas, and they come from the adrenal medulla, the adrenal gland, but then there's also paragangliomas, which are basically close to nerve ganglia from different parts of the body, and they're also still from chromatin cells. And then there's the head and neck paragangliomas, which come from parasympathetic ganglia near the head and neck, and they're from potentially a different cell type called glomus cells, but they're highly related to chromatin cells. And express a lot of the same catecholamines, but generally to maybe a bit of a lower level. And just quickly, obtaining pure samples of phaeochromocytomas is often difficult because they're from the adrenal medulla, and often you get a lot of cortex when you try and dissociate these samples. So that's something that will become more important as the talk goes on. So these tumours are functional, and they are genetically diverse. So the functional aspect means that people tend to have a lot of symptoms associated with an excess of catecholamine production, high blood pressure, increased risk of heart attack, excessive sweating and stress, things like that. And by genetically diverse, there's lots of known driver genes for this tumour, and it's also very heritable. So there's a very strong genetic component to these tumours. Something else is interesting about them. So a couple of studies have taken RNA-seq or gene expression data from these from cohorts of these cancer types, and they've grouped them into subtypes. And what they've generally found is that the subtype groupings that they come up with tend to follow the genotype of the or groups of genotypes of the tumours. So basically the genetic drivers are what drives the gene expression pattern in a lot of these cancers. The only issue with the bulk gene expression profiling is that you get information about all of the cells associated with the tumour. Like I mentioned before, sometimes that can include the adrenal medulla, the adrenal cortex, when you only want to look at cells from the adrenal medulla. And then there's also other immune cells, other tumour infiltrates that might affect the gene expression signal that you get from a bulk gene expression sample. So single-nuclei RNA-seq is a potential way you can address that. So it's similar to single-cell RNA-seq, except that it's based on the nuclei only. So you extract the nucleus from the cell and you sequence that instead of doing the whole cell like you normally would. It's able to obtain about 20 to 50% of the gene expression that you get from single-cell RNA-seq, but it's pretty essential for Phaeochromocytoma and paraganglioma because it works on frozen tissue. And samples of this tumour are hard to come by, so that's why it was good for us to use. We used the Frankenstein protocol developed by Luciano Martellotto when he was at the UMCCR. And yeah, that was highly useful to us for dissociating out different cell types. However, once you've actually run this experiment, what you get is basically this big matrix. And for each sample, you will get a whole range of cells. Each cell will have a barcode, and then for each cell, there'll be the expression of all the genes that were measured there. But it's up to us when we're analysing the data to try and figure out which cells are which based on this matrix of gene expression information for all these different cells, for all these different samples. So there's two common ways to do that. The most common is the cluster and annotate. So basically you take your gene expression from all your cells and you use unsupervised clustering to form groups. And then you label these clusters, usually by looking at genes that are highly expressed in these groups, known as marker genes. And those marker genes will be associated with known cell types, and you can then use that to figure out which cell types you have in your single cell or single nuclei experiment. The other way is direct annotation. So that basically means you take each cell and you correlate it to a known database of different cell types. And then you basically say, whichever my cell is most correlated to, that is what I think that cell is. So the first method, clustering, can be quite error prone depending on the method you use to do your single cell or single nuclear sequencing and the clustering method that you use as well. So basically this paper looked at a whole bunch of different clustering methods for single cell data, and they found that depending on the method that you use, you can get a really different number of clusters. So basically, they expected 11 different cell types in their sample, and they could end up with anywhere between like 2 and 160. So it's it's sometimes difficult depending on which method you use. There are also other factors that influence clustering. So sometimes if you have like more reads aligning to ribosomal protein genes in your cells, or you detect more or less genes in a certain cell as compared to another one, all these things can drive cells to end up in certain clusters. The SIRAH package was determined by this sort of review paper to be the best clustering method for 10x data, which is what we used. And more recently, since this has been published, SIRAH has implemented an SC transform method, which attempts to deal with some of the effects of these confounders that affect clustering. Once you've actually correctly clustered your samples, then there can still be issues depending on the marker genes that you used. So I guess if you're going to use marker genes, they have to be reliable indicators of certain cell types, and certain genes can be expressed in different cells in a wide range of contexts. So Genes such as fibroblast activated protein have been used to identify cancer-associated fibroblasts, and smooth muscle actin has been used to identify myofibroblasts, but it's been found in some studies that they actually can be identified as other cell types as well. So basically my point here is the actual genes that you pick for your marker genes, they need to be really well validated as well, which can often be difficult to do since single cells are a pretty new field. So taking all this into account, we used both SC match and marker gene annotation to try and annotate our data because we thought that would give us the highest confidence in picking up which cell types we had. So using those methods, we profiled 30 different tumours by single-nuclei RNA-seq, and we basically tried to get a good coverage of a lot of the known genotypes and subtypes of the disease, and from all of the known different tumour locations as well. So we're really trying to take like a survey of PPGLs as a whole. So as I mentioned, we used the most recent Syrah and SC transform approach to cluster these cells, and then we annotated them by both looking at marker genes and also we used SC trans. We also used the direct annotation method as well to give us additional confidence in our clusters. And what we found was that the tumours tended to cluster together, and just the tumour cells. And we also found that the normal cells, so the cells that are common in the tumour microenvironment, tended to cluster together as their own clusters. So they were made up of cells from lots of different samples, whereas the tumours were just made up of the tumours from that particular sample. So from that, we could determine sort of the overall cell populations, but we wanted to try and figure out what was going on at within given cell populations as well. So an additional challenge in doing this is that we had some ambient affecting our data. And what this basically means is that when a gene is really highly expressed in say the tumours like tyrosine hydroxylase, then it can end up at a low level or a lower level in a lot of the other samples. And the same thing with this adrenocortical marker from the normal cells, it can end up in other cells at a low level as well. So this is identified by this thing.
Yes, me. Me too.
So what I'm basically showing there is that you're seeing genes in some cell types that shouldn't be there. So we have to try and find a way to deal with this. I'll advance the slide instead of playing the video again. So the first thing I looked at as an example of this is the fibroblast population. So yeah, we were able to identify the fibroblasts overall, but trying to identify subpopulations was made difficult by the ambient effects. We could get decent separation. So this is all the cells from our normal adrenal medulla we were able to identify, but all of the tumour cells clustered more in a sample specific way. So then we tried SC match to directly annotate these cells to hopefully try and get around the ambient effects. And basically a lot of the or not a lot, but some of the cells that we annotated were based on the paper we compared against, which was a lung cancer paper. They were given, they're assigned to cells that express a certain marker gene in that paper, but we found that in our data set, that gene wasn't expressed at all in that cell type. So that was not expected. And then I did some further digging into different papers that have looked at fibroblast lineages in different tumours. And depending on the library preparation method they used or the clustering method, they ended up with different numbers of fibroblast populations, and they called all these different fibroblast populations different things. So yeah, it was really hard to figure out what we would take as our known truth for our fibroblast population, or how did we know what was real?
What is real? How do you define real? If you're talking about what you can feel, what you can smell, taste, and see, then real is simply electrical signals interpreted by your brain.
Okay, so at this point, we were pretty confused. We didn't really know how we're going to identify these cell types. So we went to the Oracle, Alicia Olschlack, for some advice on how we could better analyze this data. And she suggested that we try a pseudo-bulk approach. So what this basically is you take all your different cells from a single cluster and you sum those together, and you then treat that as just a normal RNA-seq sample. The advantage of this is that you can do standard RNA-seq differential expression, but you have a more pure population because you've only got the cells that have the cells from a particular cluster. So you can be fairly confident that, say, you're only comparing pure tumor cells with other pure tumor cells, if that's the comparison you want to make. So we did this for all of our tumor and normal cells. And what we basically showed was that we got a good clustering of tumor cells of different genotypes, so they all tended to cluster together. And this STHD cluster sort of like out sort of towards the most extreme. That's both a similar genotype, but it's also a different location. So we found that the location of the tumors actually had a little bit of an effect on clustering as well. And we found that regardless of where we took where the tumor was from, the normal cell types tended to cluster together pretty well. And yeah, we found lineages of more like fibroblast and support cells. So yeah, fibroblasts and epithelials tended to cluster out this way, and the more immune cells tended to cluster out together that way. We also found that the samples actually cluster as expected with bulk subtypes. So basically what we did is Magnus, one of the people who worked on this work, he clustered all of the known gene expression data, bulk gene expression data from Phaeos, and he then overlaid the pure populations of pseudobulk samples over that. And what we basically saw was that they generally tended to cluster with their expected genotype. And we didn't get any samples clustering in with this cluster here, which is the cortical admixture. So the pseudobulking seems to have worked as expected. And yeah, it seems to recapitulate basically a gene expression profile of the tumors that are more pure in this bulk compendium. So with this new method, we compared tumor versus normal and did differential expression to account for ambient effects of the adrenocortical cells, which were the main factor that was having an influence on our normal chromatin cells. I removed any genes that basically expressed above a certain level in that sample. So because it was the things that were most highly expressed that were showing up as our ambient effects, those are the things that I got rid of. Comparing tumor versus normal, strong differential expression candidates were observed. Some of the top genes that I saw, ISM1 and VEGF alpha, are both associated with angiogenic signaling, which is like the development of new blood vessels. And PPGL tumors, just in general, are in fact known to be highly vascularized tumors. So looking into this a bit further, VEGF-alpha is the key angiogenic signaling molecule. It was upregulated in all of the tumours, especially in the ones that are pseudohypoxic. So that basically means that they look like they don't have enough oxygen in the tumour, but in fact they do. They're just using this signalling for growth and proliferation. So then I did tumour versus normal differential expression of the normal cell types. And what I found in the endothelial cells, which are associated with the vasculature, is that they were quite activated towards new blood vessel formation. And differential gene expression showed some genes that are associated with this, and gene set enrichment analysis showed a lot of pathways associated with blood vessel formation as well. A lot of the genes that were differentially expressed were called tip cell marker genes. So they were basically markers of endothelial cells that are growing. So DLL4 is the key tip cell marker, and we found it was not very much expressed in the normal population, but there was decent expression in the tumor population. And we're also pretty confident that this was from endothelial cells because it was only expressed in that population, which is that one there in our whole data set. So the kind of like aha moment for us, if you look into angiogenesis and the processes that usually occur, tip cells are a key part of new blood vessel formation, and pericytes are this fibroblast design cell, and they actually support the vasculature as well. And what we found in our data when we went back and looked again at our SC match classification is that the majority of our tumor-associated fibroblasts were actually pericytes as well. So this kind of made sense. We had this strong angiogenic signaling, and then we had this sort of angiogenic support cell type that was showing up a lot in our samples as well. So that was like our sort of moment. And from there on, that kind of enabled us to come up with a system for refining our cell types a bit more so that we could sort of break this down a bit better. So just to confirm that most of the fibroblasts that we saw were in fact pericytes, we looked at some known pericyte marker genes, especially MCAN, and basically that a lot of the fibroblasts in that pericyte population lit up with that marker. So basically that seemed to agree with that. And then FAP, which has been suggested as a myofibroblast marker. It has been suggested as a marker of a few other things, but seemed to really show samples that were annotated as myofibroblasts in our data as well. So we could have some confidence now in the cell types that we identified. So based on that, we were able to figure out all of the cell type proportions in our different samples. So tumor samples mostly had tumor cells. Endothelial cells had a lot of tip-like cells, so that's showing that in most of the tumors, so each light dot here is a tumor, that they were growing more vasculature. Most of our fibroblasts tended to be pericytes, which again makes sense. And then for the myeloid cells, we tend to have a lot of macrophages. T cells are a bit easier to identify. I think those cell type lineages are a bit more different. And then we didn't have too many B cells, but we still tried to sum up what we had there. So basically taking these cell type numbers, I did an ANOVA just to try and see if there were any cell types that were different in one subtype versus another. And in the pseudohypoxic subtypes, we tended to, again, have statistically significantly more tip-like endothelial cells. So again, just even in cell number, that supports the idea that there's some angiogenic signaling going on in these tumors. So last thing I want to talk about cell-to-cell signaling and theranostic targets very quickly. So to figure out, we wanted to figure out which cells were talking to each other as well. So to do this, we used a tool called NAPNI, which is a network analysis toolkit for multicellular interactions. And what it basically does is it uses a database of ligands and receptors, and it compares the expression of those genes as well as the specificity to figure out which cells are talking to each other. So which cells are expressing which ligands, and then is a different cell type expressing the receptor that that ligand binds to. So it runs in a single sample. So I've been trying to combine all of that data together to get like a consistent picture of what's signaling to what. But some things that we found, for example, this subsympathetic cells is like a support cell type, and it's potentially signaling quite heavily using this IL-1 RAP receptor to PTPRD on the tumours, and that could be something that's been associated with growth. So it's possible that the subsympathetic cells are somehow signaling to the tumour and driving that pathway forward. But this is still something we're looking into. I kind of summarized all the signaling overall for the normal samples that we took and the tumour samples. In the normal samples, there's crosstalk, as I mentioned, between these chromatin cells and subsympathetic cells, and that's kind of like the biggest crosstalk. Whereas if you compare that with the tumour, you can see that that pathway is still there, and there's quite a large bit of crosstalk between the tumour and the tip-like endothelial cells, which are the ones that are driving the new vasculature. And they're also talking to the pericytes, which is the support fibroblast. So again, that kind of supports what we'd seen earlier. Okay. Theranostic targets are basically, theranostics is like a method that targets receptors. So you bind a radionucleotide to a ligand that targets a receptor that's overexpressed by the cell type. And then that can be used to both image and treat the tumor. So that's why it's like diagnostic and therapeutic. And this has been done in neuroendocrine tumours before. So this is work that Rod Hicks does. And we want to find some new potential receptor targets that he could potentially use in his work. So from our tumour normal analysis and from looking at what was highly expressed in the tumours, we've identified this gene GPR139, which is a receptor with no known ligand in this circumstance, an orphan receptor. And it seems to be pretty highly expressed in the pseudohypoxic tumours, especially the VHLs. So that's potentially a target for that sort of theranostic method. We wanted to make sure that it wasn't expressed in a lot of other places for off-target effects, and it seems to be fairly specific to PPGLs. So this is from the TCGA, and basically the highest expression of GPR139 is in the PPGL tumours, and there's a lot of tumour types where it's not particularly expressed. The next highest is glioblastoma, which is sort of related disease. We also looked in the GTEx database for places that it was expressed, and there's a little bit of expression in the brain, but for the majority of cell types, there's not much expression. So this is probably a good target to look into further. Okay, so what I hope you've got out of the talk, when you're doing a single cell experiment, you should use SC match or some sort of similar approach and marker gene expression. And we use this to identify our tumor biology at the cell type specific level. We found the activation of endothelial cells is supported by the known biology in these tumours, and it makes sense in terms of the cell-to-cell signaling we saw as well. Seems like angiogenesis is key to PPGL progression, and that's supported by the tumour microenvironment. And we also found that GPR139 is a subtype-specific theranostic candidate. What we need to do is finish the cell-to-cell signaling, start to validate the theranostic targets, and finish writing the paper is probably the most important thing. Just like to thank everyone at the UMCCR, Richard, who has headed this project, and Magnus, who is the other bioinformatician who's done a lot of work on this. I'd like to thank Luciano and Fernando for their help with running the single nuclei analysis and getting us the data, and then all the other people who have had either input or part to play in this as well. Also, I'd like to thank you for listening, and the rest of the radio lab as well. That's us. And does anyone have any questions?
Thanks, Andrew. That's a great talk, and always very entertaining to see the themes that you have with the matrix here today. Just like to point out, I guess, as well, I guess my fault for not giving you an updated acknowledgement slide, but also acknowledgement to Al Forrest and Rui Ho over in Perth at the Perkins for their contribution to NAPMI analysis. So I'll open up to questions. Is anyone got any questions to send in? So Andrew, maybe while people are thinking of questions, can you say anything about the general quality of the single nuclei data compared to single cell in terms of variability, maybe the amount of ribosomal RNA, etc., that you're seeing in those samples?
I did, well, the QC analysis was definitely more magnificent side of things, but I don't think you see as much, well, you shouldn't see any ribosomal RNA really in a single nuclei sample. I guess mitochondrial as well, we did see a little bit of, but that's usually an indicator that the data is, or that the cell is not great. But yeah, you really shouldn't see any of that in nuclei sample. One thing, I guess, that we did see, I guess people will be aware of analyzing nuclei data, is we saw like a huge amount of nuclei-specific protein, which is obviously to be expected, but that's just kind of something to be aware of when you analyze this data. There's like a nuclei expressed gene, and that was easily our top expressed gene in all of our samples.
Okay, we've got a question from Lavinia Gordon. Can you remind us why you did single nuclei analysis and not single cell?
Yeah, that's just because you can be used on frozen tissue. So because the disease is so rare, it's not really possible to get fresh samples. So we have to use archival tissue for this sort of work.
Another question from Lavinia. Can you discuss the reference data sets with SC Match? Would it be worthwhile generating an in-house reference set?
Yeah, it could be. So this is what Magnus did, but what he basically did for that was he took, it was a lung cancer paper, which was published pretty recently, I think it was this year. And they were pretty good on their cell type annotations. So he basically just took their clusters and he took the average gene expression for a particular cluster. And he used SC Match using that as a reference database. So yeah, so in some circumstances that worked, like the perisite identification, but in other circumstances that didn't work so well. So if you did that for a range of different papers, I suppose that you could get a lot of good information about which cells from which papers your data was correlating to. So yeah, that could definitely. I mean, SC Match also has its own database, but yeah, you can certainly expand on that if you have papers you want to compare against.
And just a comment from Jay Doughton. Great presentation. Love the matrix analogies, which worked really well.
Thank you.
Another fan. That's pretty much the end of the questions, and I think we've pretty much come to the end of our time. So thank you, everyone. I think Andrew, just